My name is Laurence Reid. I'm the Chief Executive Officer at Decibel Therapeutics, and I'm very pleased to be here today to have the opportunity to share Decibel's strategy and tell you about programs in our pipeline. This is really our summary of Decibel on one slide. We're building what we think can be one of the leading companies in the field of novel therapeutics for hearing loss and balance disorders. There are massive unmet needs globally today in both hearing loss and balance disorders. There are no approved therapies for either of these very significant and debilitating conditions. And there's been very significant lack of investment in these fields, whether by the pharmaceutical or by the financial industry historically. Under the broad set of opportunities for treating hearing loss and balance disorders, in particular, we believe there's a very exciting set of opportunities to bring gene therapy to the inner ear and to deploy the knowledge that others have built around deployment of gene therapy over the past 10 to 20 years. On top of those advances, Decibel is using its integrated platform, which really deploys single-cell genomics to build a pipeline of precision gene therapies, by which we mean gene therapies where we control the expression of the transgene selectively in the target cells where the gene therapy product candidate is designed to elicit the biological and pharmacological response. We have a focused pipeline. Our initial candidate products are for monogenic and congenital forms of hearing loss. Our longer-term pipeline is focused on building regenerative medicines that will regenerate hair cells in both the cochlea for the treatment of hearing loss and in the vestibule for the treatment of balance disorders. We've built an outstanding and experienced team, bringing together experts in the field of the biology, pathology of the inner ear, together with extensive drug hunting and company building experience in other disease areas. We've been working for many years now with an outstanding cadre of biotech investors who share our vision that now is the time to bring innovation to therapies for these very important medical fields. This is Decibel's pipeline in the later half of 2021. I want to note that we retain 100% of the commercial rights, global commercial rights in this pipeline. Pipeline comprises three categories. At the top, we have our gene therapies for congenital monogenic hearing loss, with our otoferlin program being the vanguard program in this set of product candidates. Second, we're building a pipeline of gene therapies for hair cell regeneration. Our initial product candidates will aim to restore function of the vestibule by regenerating hair cells in that organ. And then also we work in terms of regeneration of hair cells in the cochlea with a focus on different forms of acquired sensorineural hearing loss. Thirdly, at the bottom, our otoprotection therapeutic DB020. And note this is a small molecule, so a different type of strategy and approach for this product. And we're going to take this strategically down a different path through some type of partnership after we are able to share our preliminary interim analysis from our phase 1b study in the first half of next year. I note that the monogenic congenital programs are partnered with our colleagues at Regeneron, a very interesting alliance where we collaborate with Regeneron on the research and development of these product candidates. But Decibel retains the full commercial rights, and we compensate Regeneron for their participation and contribution to the program with a series of royalties when the products get to market. In terms of these two fields of hearing loss and balance disorders, as I mentioned, very significant unmet global needs today. But importantly, these fields now are lending themselves to approaches of precision medicine where we're able to begin to disaggregate these massive populations. For example, the use of genetics to define monogenic forms of hearing loss. And on the balance side, although there is no definition of balance disorder populations through genetics today, we are able to focus on particular acute forms of balance disorder, for example, the bilateral vestibulopathy, which often has a very clear molecular etiology that we're using to focus on particular sets of patients. So our strategy overall is to deploy our platform, which integrates single-cell genomics and bioinformatics applied to the inner ear and building a database, which we believe is the largest in the field in terms of molecular profiles of the inner ear, to build a pipeline of precision gene therapies where the transgene will be expressed selectively in the correct cells of the inner ear, together with a lot of experience and expertise now in the inner ear itself, its biology, its pathology, the conditions that arise from that pathology, but also how do we deliver different types of drug candidates to the inner ear safely and effectively, including both small molecules and gene therapies. Those technologies come together in a broad umbrella to restore the functionality of hair cells, whether by gene transfer to complement mutations that manifest themselves in the functionality of the hair cell, or to regenerate hair cells to replace hair cells that are dying or perhaps have been killed over the course of one's life. That biology is then aimed to come together to treat either rare monogenic conditions on one hand, for example, in the hearing loss area, or the more common acquired forms of either hearing loss or balance disorders. Just briefly on gene therapy itself in the inner ear, we believe that gene therapy and the inner ear are a remarkable marriage of a technology and an organ, and that this is a very promising modality for treating conditions that arise from pathology in the inner ear. And I note that, of course, there are very strong parallels between the ear and the eye. Advantages of the inner ear include the fact that it's a small enclosed compartment. We're therefore able to deliver very small quantities of drug. Illustrated on the right, we think we're working with quantities of drug that are 3 to 4 orders of magnitude less than those required for systemic forms of gene therapy. We have a very high ratio of the drug product to the target cells. Secondly, the ear enjoys a significant degree of immunoprivilege, and like the eye, and therefore we think there's good potential for improved tolerability relative to some of the challenges with systemic gene therapy. Because of the enclosed nature of the inner ear, the drug generally is retained in the inner ear, and there's very negligible systemic distribution of the drug candidate into the systemic circulation. Again, we believe creating opportunities for improved tolerability. Thirdly, we're targeting one fixed location via an established surgical approach. We use the surgical approach, which is deemed by KOLs to be optimal today for reaching the inner ear. It's the same procedure that they use on a daily basis to implant cochlear implants into the ears of both children and adults. So it's a well understood route to access, and we're accessing a single location in the inner ear. And lastly, the cells that we're targeting are non-dividing, which we think gives us an optimal chance of durable expression over the longer term. So these are all, we think, very important advantages of gene therapy for the inner ear, and that give us a lot of excitement in the promise of the pipeline that we can build using this remarkable modality. Quickly on our pipeline now, gene therapies for congenital monogenic hearing loss. Our lead program, DB-OTO, aims to treat hearing loss caused by congenital genetic loss of otoferlin. Otoferlin is a calcium sensor. It functions at the base of the inner hair cell. Children born with biallelic mutations in otoferlin suffer from a congenital profound hearing loss. They're detected by newborn, followed by genetic testing. We think there's very significant opportunity in this focused orphan disease. Other companies are also working on this product. We believe our product is differentiated. It's a precision gene therapy with selective expression of otoferlin in the hair cells. It's delivered via a standard surgical procedure that I mentioned. And lastly, we've invested a lot of time in relationships with institutions such as the Ramon y Cajal in Madrid, who have access to what is believed to be the largest characterized cohort of patients known in the hearing community today. These are our mouse data. We're using a dual hybrid system to deliver the otoferlin gene. And what we see in our animal studies is that if you treat an animal that is lacking otoferlin, a complete lack of red expression, and has a flat auditory brainstem response due to the mutations, after treatment within a small number of weeks, we see instatement of normal expression of otoferlin selectively in the hair cells. And now we've instated in these animals essentially a normal auditory brainstem response. So we're seeing the selective expression of our transgene and a robust functional recovery. We've been able to quantitate that we need to transduce about 20% or more of the hair cells of the inner ear in order to achieve this functional response. As we move to non-human primates on our way to the clinic, we're now in those types of studies. We see excellent reproduction of the molecular selectivity of expression as we move to non-human primates, and we're getting transduction of hair cells in the inner ear of the non-human primate at a level of 75 to 95% of the inner hair cells, well above the functional threshold that we defined in our earlier rodent studies. Moreover, as we've done more extensive studies on durability of response in our rodent models, we see an advantage of our cell selective promoter. It appears to give a much more durable response compared to the constitutive promoters that are often used historically in the field and which are used in some of the competitive products. Our program is marching forward now towards the clinic and to first patient treatments in 2022. We had an excellent introductory meeting with the FDA at the end of last year, gave us a very clear path to the clinic in terms of the preclinical animal studies, and also an excellent understanding of a proposed phase 1/2 trial design. Lastly, on the manufacturing side, we collaborate with our colleagues at Catalent, and they'll help us produce all the materials now required for the preclinical and clinical studies. The other gene therapy program for monogenic forms of hearing loss that we've announced so far is our AAV103 program. This aims to restore hearing in individuals with GJB2 deficiency. GJB2 is the most common mutant form of hearing loss. Over 250,000 patients are believed to exist in the US and major European markets. And our approach is to deliver AAV103 for expression of GJB2 selectively in the constellation of support cells that surround the hair cells to restore hearing in these patients. Second part of our strategy for the longer term part of our pipeline is gene therapy for regeneration of hair cells. Our strategy is to convert the support cells, the non-sensory cells that are interdigitated in the epithelium between the hair cells, to convert those support cells in situ into hair cells to replace hair cells that are lost over the natural course of life. We're doing this by identifying key reprogramming factors using our single-cell genomics platform, and then using our proprietary toolkit of cell-selective promoters to express those reprogramming factors in exactly the right required cell types. In the vestibule, as I mentioned, we're looking at treating patients who suffer from bilateral vestibulopathy, which is a particularly profound form of hearing loss. We're aiming to regenerate both type 1 and type 2 hair cells in the vestibule in this program. And again, looking to use our cell selective promoters to express the right reprogramming factors in the support cells to drive the conversion of those cells to hair cells. An example of this is our 201 program, where we combine 801, a transcription factor known to be critical for hair cell development, together with a second reprogramming factor. And we think the combination of those two factors is going to let us regenerate both type 1 and type 2 hair cells in the vestibule. Importantly, we think we're going to be able to deliver both of these factors together in a single gene therapeutic. We have a very similar strategy for the cochlea, where we're looking to drive the regeneration of both inner and outer hair cells by conversion of the supporting cells. Very similar strategy where we use our genomics platform to identify the factors, and then our gene therapy platform to express those factors in the correct cell type. Lastly, our otoprotective therapeutic, DB020. This is a small molecule that's administered to the inner ear locally for the prevention of the very profound hearing loss that is induced by cisplatin in the course of cancer chemotherapy. Over 250,000 patients today are still treated with cisplatin around the developed world, with very significant risk at high doses of developing hearing loss. Our product incorporates a validated mechanism, a small molecule sodium thiosulfate, which is a natural metabolite, and is able to very efficiently inactivate cisplatin locally through our proprietary formulation and a procedure to deliver the drug directly into the middle ear, and the small molecule then is transferred to the inner ear in advance of the chemotherapy, providing a very significant degree of protection against cisplatin-induced hearing loss. The program is in clinical trials. We completed a phase 1 study in 2019 and are now involved in a phase 1b proof of concept in both Australia and the US. And we expect to report on an interim analysis of this study in the first half of 2022. So I'll wrap up. Hopefully I've had a chance to both convey the huge set of opportunities that we believe exists in the field of hearing loss and balance disorders, and a little bit about why we think gene therapy can be such a profound opportunity to develop novel therapeutics for the inner ear, and how the platform, the single-cell genomics and precision gene therapy platform in which we've invested at Decibel over the past several years gives Decibel an outstanding opportunity to bring novel medicines to the patients in each of these incredibly important fields of modern medicine. Thank you for your time and interest in Decibel.